you are in the world. I'm Amy Keating. I'm the president of the Protein Society, and I'd like to welcome all of you here today to the webinar on understanding proteins in membrane environments with Cryo-EM, which has been organized and will be moderated by Brian Dwyer from Pharma. I'll just make a few announcements here at the beginning about the Protein Society and some of our activities. Actually, let me go back. So for those of you who don't already know this, the Protein Society is an international organization. We have members in approximately 30 countries. And our two major activities are that we run an annual meeting, about which I'll talk to you about that more in a minute. And we also publish the journal Protein Science, to which I hope you'll all consider submitting your work. The journal is in a period of transition because longtime editor-in-chief Brian Matthews is stepping down after 15 years of dedicated service, and he will be replaced next January by John Currian, who will be taking over. So we're very excited about this and would like to welcome John. I've highlighted here just a few articles from Protein Science that are on topics related to today's webinar. And I hope that those of you who are listening will also consider submitting your work in your papers to Protein Science for consideration. Let's see, so over the past year during the pandemic, in lieu of in-person meetings, we've been running webinars like the one we're doing here today. And these have been really a great success. Many of the talks on the topics that are listed here were recorded and are available on our website if you missed them and would like to go back to that. Our fourth webinar was particularly special. This was organized by Bill Clemens and the Protein Society DEI committee, and this featured a panel of our Protein Society stars who spoke about issues of diversity, equity, and inclusion in their own lives and at their own institutions. And that was recorded, and you can find their remarks on our website if you missed it. So let's see, another initiative of our DEI committee is our Diversified Protein Science website. This includes a database we've put together of protein scientists from underrepresented groups. And I encourage you to register yourself here if you're a member of an underrepresented group. And also I encourage everybody to use this as a resource when they're planning meetings or staffing panels or looking for leadership candidates. So this July, we'll hold our 35th annual symposium. And unfortunately, this will be run as an entirely virtual event, although it's going to be spectacular nevertheless. We have a phenomenal program that was put together by Planning Committee Chair Jeannie Hardy and her team, who you can see here. This meeting will include 12, I believe, plenary talks by both our 2020 and 2021 Protein Society Award winners, who are a spectacular group that you can see here. And there will also be lots of terrific invited and contributed talks and live poster presentation opportunities and community events that will provide opportunities to interact and meet new people. So registration for that is currently open. It's very affordable, and there are also a number of awards you can apply for to subsidize the already affordable registration. And there's still time to submit an abstract for a junior or senior contributed talk or a poster. We hope that you'll do that this week if you haven't already, although late breaking posters will still be considered up through June 1st, so you can send in your latest, greatest results. Okay, so last few things. As usual, we have a few open opportunities for our members. First, our annual symposia feature member organized sessions, which give you an opportunity to pick a topic and invite your own speakers. And so our call for proposals for that is currently open for our 2022 in-person San Francisco meeting. So we encourage you to consider submitting a proposal for that. Also, we're still considering proposals for member organized webinars like the one that Brian is hosting here today. So if you have your own idea for a program and speakers that you would like to organize with us, please send that in using the information at the website. And so with all that, I will hand things over to member and webinar organizer, Brian Dwyer, who will explain the webinar protocols and introduce today's topic and speakers. So thank you, Brian, for bringing us all together today to hear about this interesting work.
Great, thank you. Let me just share my screen. And just a brief comment about the questions. Again, I'm Brian Dwyer. I'm the moderator for the webinar this afternoon, and welcome. And basically, for questions, when you get to the Zoom, you can go to the Q&A part. So I encourage you to write any questions you have that come up during the presentation. And if you can, if you put the first name of the presenter for that question, that'll help me, because I'm the one who's going to be going through all the questions, and I'll bring them up to the appropriate speaker when the time fits. And again, for the time slots that we have, we can't get to all the questions, but I'll try and pick a few that are quite interesting for the speakers, and we'll do our best to get to them. And again, after this webinar is over with, there is the opportunity for the speakers to stay on, and they can engage the questions themselves. So there's that as well. And again, even if you have questions that aren't answered, I encourage you to go to all of these panelists' lab websites. They're also on social media, LinkedIn. You can connect with them that way as well as myself. So again, my email here is brian.dwyer@pharmarun.com. So any questions you have about the webinar or anything going on, I'm happy to connect you or get information to you as well. So a little introduction about myself. So I'm at Pharmarun, which is a CRO company, and I'll talk a little bit about that a little more in a slide down the road. So I've been there for about 10 months. I'm a business development manager covering a lot in the US and some in Europe. I'm in the biologics department, so I'm all about protein-related services. So it makes sense that I'm there because I've spent the past 22 years in biotech and pharma doing protein sciences. I've been in small startup companies all the way up to large pharma companies. We're usually focused on protein production, protein purification, and characterization. And a lot of protein biochemistry and biophysics has come along. And I'm now in an MBA program, so I was interested in the business side of science, so here I am. I just want to give a little quote that I like. It's you miss 100% of the shots you don't take. So I encourage anyone in science or in life, if you got something you really want to do, just go for it. Don't worry about falling down. You'll get right back up again and keep going. I also produce a very small podcast called Transition States Podcast, and it's all about interviewing people in biotech, biopharma. So anybody out here in the audience wants to be interviewed, just let me know. And it's really to give career advice to people like the grad students at UMass Lowell, where I've coached a little bit. And most recent addition to my family is a dog named Thor, which has really kept us quite busy the past couple months. But that's just a little background of myself. And I want to do a big welcome and thank you to everyone. There's a lot of welcoming and thanking to go on here. So I really want to welcome you all to the 7th webinar. And I want to thank the Protein Society, and I'll get a little bit more about that. It's a wonderful platform for webinars, and as you will see. And I want to thank Pharmarun for sponsoring the webinar. And obviously our scientific panel, they're very easy to work with, wonderful people, and it's been really great pulling this all together with them. And most importantly, I thank the audience. I really want to thank everyone for registering and taking the time to be here this afternoon, and it'll be well worth it. So again, a little more on the Protein Society, membership, and things that are going on. So it's a wonderful society. If you haven't been a part of it, you should probably join. And I've been associated with them a long time. I think my very first professional symposium was with the Protein Society many, many years ago. And I try to make as many of them as I can. And also in regards to webinars, I encourage anyone out there who has an idea about a webinar, you have some scientific friends that you could easily pull together. I encourage you to go for a webinar or to utilize this platform, because it was very easy to do. I mean, it was working with the Protein Society is wonderful. And I'll just say, like Raluca, it's fantastic to work with, got some good feedback on the topic, took you through the whole process. And her team is wonderful. We had some questions. They're very fast to deal with things. So anyone out there, I'm just letting you know, I just went through it myself. It's a lot of fun. It's really easy. And it's a great platform to get some information out there to a lot of people. So I also encourage people to consider membership or to invite someone to become a member and join a committee. So why do I say all that stuff? Because I actually done these things myself. So I'm part of the education committee and the membership committee. And again, it's a lot of fun. The amount of work you do is very reasonable, and it's really only comes up a couple times a year. So really go for anything you can with this society. It's really well worth it, and you get a lot of good relationships out of it. And to Pharmaron, where I'm currently is my employer, very proud to be a part of them. And we're very proud to work with the Protein Society and to sponsor this webinar. So we're a fully integrated pharmaceutical R&D services platform. And what I'll focus on for this particular webinar and where I'm housed really is in the biologics department. So we're all about protein production, expression, purification, and characterization. We also offer crystallography services and cryo-EM services. We've entered into a biologic CDMO space as well as cell and gene therapy lab services most recently. So anyone has any questions about this, please reach out to me. I'm happy to take any questions, and we're doing great work there. And I'm really proud to say this, that really the work comes into customizing our services to our clients. And that's another reason why I'm very proud to be part of Pharmaron. We really go out of our way to really get our clients what they need for the work they need done. So now to our panel, I really like to thank all of them. Doreen, Malfu, and Mahmoud have been wonderful to work with. And they have wonderful scientific stories to tell. And they have many more than they have time for today. And I just want to introduce Doreen. She's part of the unit on structural biology. She's part of the Division of Basic and Translational Biophysics at the NIH Center in Bethesda, and mostly housed in the childhood disease area. So also very shout out to her. She just recently started her own lab there. And if you check her out on Twitter, she actually posted her picture of swearing in. So I think it was your back deck is my guess. So that's very exciting for her. And introduce Malfu. He's associate professor at the Department of Cell Biology at Harvard Medical School. And he has a wonderful story to tell about ABC transporters. All of these talks are, proteins within membrane environments. And we'll close out with Mahmoud, who is an assistant professor of medicine at the Department of Medicine, the Brigham and Women's Hospital and Harvard Medical School. And he'll be talking about an activated GPCR complex with its G protein in a membrane environment. And to start us out will be Doreen, and she'll be opening the door for us really on a historical and educational and sort of a development perspective on cryo-EM and the field's progression till today. So with all this, I thank everyone for being a part of this, and we'll start off with Doreen. So Doreen, if you could take it from here, that'd be great.
All right, let me share my screen. Can you see my my screen now? I hope you can.
It's in the double right now. You got to switch over.
Oh, okay, sorry. Let me try that one more time. Desktop 2, share.
There you go.
And then I go into my presentation mode. You can see my slide now, okay?
Good. Looks
Yeah,
good.
okay. Thank you so much, and sorry for the little hiccup in the beginning. Yeah, thank you, Brian, and then also to the Protein Society for the kind introduction and inviting the three of us here to talk, and hopefully start a nice discussion afterwards with all of you, how we can now study membrane protein complexes in their lipid membrane environment by cryo-EM. And I offered to give an introduction on the most common methods currently currently used in Cryo-EM and show you examples from complexes I have worked on, as well as examples from other groups. But before I start, I thought I'd give you a quick overview of the various projects I worked on in my career so far, so you kind of get a rough idea of my background. So I'm a structural biologist, and I've always been fascinated by membrane proteins. Yeah, I cannot believe that it's been more than 15 years already that I've been looking at membrane protein structure and function. Initially, I used super tightly reconstituted membrane proteins into lipids at a very high protein to lipid ratio, forming 2D crystals here, to using low resolution methods like single negative staining, single particle EM. Back then, the only method really back then to look at larger complexes at high resolution was X-ray crystallography. So I was lucky enough to be able to solve some membrane protein structures using that before the entire field opened up in Cryo-EM with all these new technology developments and the new direct electron detectors and more stable microscopes, imaging, processing software development that really opened up the field to looking at not just really large protein structures, but smaller and smaller protein structures, including then membrane proteins, initially in detergent or amphipoles, and later on, now you will see in our talks, in a lipid environment like lipid nanodiscs or liposomes or even in situ. So membrane proteins have always been challenging to work on, and here on the left you can see a statistic of that really shows how challenging it is to look at membrane protein structure. So these are PDB entries of membrane proteins in orange here versus all proteins by year, and you can see that the lack of protein structures basically is a huge indication that it's very challenging. And why is that? Membrane proteins are incorporated in lipid membranes, and for us to study them, we have to take them out of their lipid environment. And the most common way of doing it, or of initially characterizing membrane proteins, was using detergents that solubilize these membrane proteins, but they are associated with protein instability, poor compatibility with structural as well as biophysical studies, since they just do not accurately mimic the native environment of membrane proteins. Here I'm showing you a traditional membrane protein purification for structural biology using detergents, which many people still currently use. But I want to say that even the mildest detergents tend to decrease protein stability while increasing protein aggregation. And in this representation here on the left, you can kind of see why. Initially, the membrane protein is nicely embedded in a lipid bilayer, and once detergents are added, they start to delipidate the protein, and the end products are the detergent solubilized membrane protein, if your membrane protein is stable enough, and then detergent lipid micelles and detergent micelles as well, you can see here. And here I actually want to make a big point on these detergent micelles, which increase the background of negative stain or Cryo-EM images a lot. And many people don't know that the background of these detergent micelles really is a challenge in our field, and many people don't know how much detergent they actually have in the sample, because they concentrate these detergents when they concentrate the membrane protein. And so far in Cryo-EM, when you look at protein structures in detergent micelles, the protein concentration is pretty high that you need to produce or make these Cryo-EM grids. And for that, you typically have to concentrate, and you do concentrate the detergent as well. And And I want to highlight one of the papers that actually looked at that very carefully from the group from Arne Möller's group, who just looked at detergent at different concentration, common detergents that are used, and pure detergents, put them on EM grids. In this case, it's negative staining, but you can see all these little structures that could potentially look like your membrane protein are actually just really empty detergent micelles. And this is every single membrane protein project that I've worked on, starting by quickly screening these samples by negative staining. I've seen these detergent micelles, and I always encourage people to look at their buffer controls that they treated the same way that they treated their sample, including concentration steps, to really make sure you're not looking at just detergent micelles, but your protein in detergent, if you're still using detergent. But this gets me to my summary slide basically already, and then I'll show you a lot of examples of an exciting news of how you can actually keep your membrane proteins in some sort of lipid environment and use it for Cryo-EM to look at the structure. So most Cryo-EM structures of membrane proteins so far have still been solved in detergent micelles or amphipoles. But recently, I'm super happy to see a shift in more and more structures coming out that are in some sort of lipid environment. If you know what lipids are needed or bound by your target protein, you can simply just add some of these, which may stabilize your protein a bit, and we call them bicelles, and we've done that in our field for X-ray crystallography already. So the moment you know something is important and relevant and for your protein, just add it during the purification, and that might already help. The most common method these days is reconstituting your membrane protein into lipid nanodiscs using MSPs, standing for membrane scaffold protein, which are apolipoprotein based, and a whole library of different constructs and different lengths of and properties forming different sized discs is now available, thanks to the great efforts of many different groups, including the Sligar and the Wagner group and many other groups. And we will hear more in the next talk too about circularized membrane scaffold proteins. Another similar approach is using saposin lipoprotein, short Salipro. Saposins are also known to be modulators of lipid membranes, and with their lipid binding properties can be used to form lipid nanoparticles. Another similar approach not mentioned in this review here are peptidiscs, which use similar to these apolipoprotein like scaffold proteins. Just the sequence is reversed, and they engineered it a little bit more to have a higher lipid affinity, but they're much smaller, and multiple copies of these can form around the membrane proteins, including lipids, and form these peptidiscs. All these three disc-like approaches still require the initial solubilization using detergents. That is why I'm so excited about the so-called SMALPs, which is here on the bottom right, or I call them native lipid nanodiscs, which use amphiphilic copolymers, such as the most commonly known is the styrene malic acid. And these copolymers spontaneously insert into membranes and break them up into lipid nanodiscs, and basically not requiring the solubilization step by detergents, and basically leaving a patch of native lipids, lipid membrane around with that hopefully never destabilizing them. Another common way of analyzing the function of membrane proteins, looking for example at transport of molecules across membrane, we use the liposome system, where we reconstitute again purified proteins into liposomes, and there has been some exciting news about how we can also use that in Cryo-EM and solve high resolution structures in liposomes. And if I have time at the very end, I will talk to you about in situ structural biology, which are still kind of in the lower resolution range so far, but you can look at subcellular fractions or even cells that you thin down using fit milling. So really exciting approach. But coming back to the first one, that the most common approach, membrane protein reconstitution into lipid nanodiscs, you basically, as I said, for these three methods that I mentioned, you still start with membrane proteins and detergents, but the sooner you can put them into this lipid environment, the less delipidation you will have, because over time you just delipidate more and more. So you incubate your membrane protein and detergent with solubilized lipids, add your scaffold protein or your saposin lipoprotein or your peptidisc peptides, and then you start removing the detergent using biobeads is the most common one, but I will tell you in a moment, be careful with that. You can use dialysis, which takes maybe some time, gradient centrifugations, dilutions, whatever works for your protein, try it, and then you basically form a lipid nanodisc. The first time I worked on a nanodisc sample was back in 2014, 15, I think, where we looked at magnesium channel core A that we back then solved in different conformations in a DDM detergent mice cell by Cryo-EM. And here we wanted to check if we can also observe these asymmetric conformational changes in the lipid environment, which we saw in detergents. Which in the end we did. But one observation we initially made was that even though we selected a belt protein or MSP that forms a roughly 10 nanometer nanodiscs, we could classify these particles that we got in the end into classes with multiple different sizes of discs, with this one probably around 10 nanometers, but we noticed there were particles having a much smaller nanodisc, which was a little bit of a concern initially. And here we used biobeads to remove detergents, and we left it overnight initially. And that was probably something I would carefully advise people to not use too many biobeads and to not have these incubations for too long. Otherwise, we believe that these biobeads may pull out some additional lipids that are actually inserted into these lipid nanodiscs, just to keep the nanodiscs as large as you can. In 2016-17, I looked at a voltage-gated potassium channel in lipid nanodiscs. The reason for it back then was to look at the interaction of channel activating toxins in complex with the channel, which they had found to interact with the lipid bilayer. So we really needed that lipid environment. And we chose in this case a slightly larger disc size. As I said, these belt proteins come in various different sizes, so you can really pick and choose. And Chen Yung did a fabulous job doing all these initial tests, which are required initially to find the perfect lipid to protein ratios to form these discs. And then I highly recommend looking at these samples, even if they look good on gel filtration profiles and SDS gels and native gels, to really look at your sample, no matter what disc technology you're using, to look at them with negative stain. And this is what we've done here. Membrane proteins in discs are so easy to recognize. You can always see these little rod shape like particles. And with this, you really know how good your solubilization is, how good your reconstitution is, how homogeneous your sample is. And in this case, if you spend just half an hour, you get an image like that. If you spend some more time collecting data, you can run 2D classification. And in this case, it was super helpful to us to see that the sample looked pretty good on a single image, but then when you actually analyze it, this channel has an alpha subunit that's embedded in the membrane with some sort of little soluble domain, but then it also has a beta subunit on top. And we could see that the subpopulation of our particles actually lacked the beta subunit, so the beta subunit dissociated from it, We also, from negative stain, really a simple short experiment, we saw that the soluble domain was slightly flexible compared to the membrane domain. And we saw that the protein wasn't always in the center of the disc. So these were challenges we knew we had to overcome during processing. And that I knew, so I knew what to do once we had high resolution data, having Cryo-EM data. So again, this is a Cryo-EM image. Again, for membrane proteins, it's so easy to see these lipid discs, and if you start seeing densities inside your disc, that's a very good sign. You have thin enough eyes and enough signal to noise, so you will be able to solve structure. As I said, we applied what we learned from negative staining about these subpopulations of particles missing either the beta subunit or having this flexibility, and used extensive classification, masking, and focused refinement to come from, an average, structure with many low resolution regions to using signal subtraction and classification to really increase the local resolution in both the soluble domain and the membrane domain. But enough of my examples for now. The most well-known of one of the first high-resolution membrane protein structures in lipid nanodisc was the tryp channel, of course, that I think everyone knows, resolving some tightly bound lipids, which was super exciting back then, that you can see here in blue. But looking at structures in nanodiscs, there were a couple already published before, and of course, at lower resolution. But I always love to show, especially this example of this membrane part of the V8PAs, which was, I think they'd never published it in a paper, but just based on improved technologies, they could improve the resolution to such an incredible amount of information just after using, the new technologies. And here a couple more examples of different structures solved in lipid nanodiscs, and I really look forward to hearing more about the recent structures of ABC transporters and GPCRs from Maufu and Mahmoud right after this. So super exciting too these days. So these were examples of nanodisc structures using these membrane scaffold proteins, but what about the systems using the saposin, lipoprotein, and the peptidiscs? So they are maybe less well known, but I thought I'm including them here. So especially in the last few years, some labs have successfully used the saposin lipoproteins. And in the case of the calcium uniporter, for example, here on the left, you can really see how 6 of these saposin molecules wrap around the lipid membrane part of the protein. Here on the right, I would like to highlight a recent example of Misha's group on the various conformations of the serotonin receptor, which are known to be modulated by the lipid environment. So it was important to solve these structures in a lipid environment. And compared to previously published conformations in Detergent, the lipid bilayer stabilized the receptor in a more tightly packed coupled states, and besides finding an asymmetric stage, which was really exciting to me, which suggests an asymmetric activation of this receptor, they could also resolve the agonist bound receptor conformation, which adopts a wide open pore capable of conducting sodium ions. So very exciting, slightly different technology, maybe slightly more flexible, and I encourage people to just try this as well. The Tom Valls group has published two higher resolution structures from Cryo-EM using these peptidisc peptides. And again, this group can really show how these peptidisc peptides wrap around the membrane part of, in this case, an ABC transporter, and this one is a mechanical sensitive channel. Different ways of stabilizing these, but again, this is one of, besides the typical MSPs, these two are out there too, and seem to be working really well. But as I said earlier, all these 3 disc-like approaches still require the initial solubilization using detergents. That is why I'm so excited about these so-called SMARTSO native nanodisc, which were developed about a decade ago. And by now we have a variety of different amphiphilic copolymers available to synthesize either ourselves or purchase them commercially. The main problem I'm hearing, especially about the polymers available commercially, is that they do not solubilize extremely well, and that they don't form homogeneous sized particles, and that it's not so easy to work with them using size exclusion chromatography. But I would still like people to keep trying and experimenting with them, since I think this would be really a huge difference to keeping most of our fragile membrane proteins that we have no access to right now, to keep them stable and do structural biology with them. And on the top here, I'm showing you how these copolymers spontaneously insert into membranes and break them up into lipid nanodiscs, which can then be further purified. Again, no solubilization step by detergents is needed, and basically leaving a patch of native lipid membrane around, which with that hopefully never destabilizes membrane proteins. And here in the bottom, I don't know if I collected them all, but the first structure I knew of is ACRB, which was solved at around 3 angstroms, the bacteria multi-drug exporter. We have a super complex of complex 3 and the cytochrome C oxidase, both from bacteria using these copolymers, in this case, styrene malic acid. And 2 recent examples from the Eric Gore lab, looking at these were actually really exciting, because these examples are from bacteria, but Eric Gore's lab expressed these proteins in mammalian cells. So, and then he used 2% styrene malic acid, incubated for an hour, and basically solubilized these out of these membranes, and was able to purify and then solve these structures at quite high resolution and in many different conformations. So it's super possible to do it. I know it's tricky to work with them, and I would just encourage people to talk to the people who have used them, check which companies they ordered that from, or how to synthesize them, and really give it a go, because it's so important to keep that native environment around. I've mentioned membrane protein structures in liposomes, and I wanted to show you a few examples here. Misha again has looked at the serotonin receptor in tightly reconstituted proteoliposomes by cryo-electron tomography and subtomogram averaging, getting around a 12 angstrom map. But below, here you have more single particle approaches. With the first one, I remember seeing from Fred Zigwer's lab at Yale, looking at the potassium channel in liposomes already 10 years ago. I added this example of a macrophage expressing gene 1, which is not a typical transmembrane protein complex, but it can form pores in target membranes like bacteria upon acidification. And in the presence of liposomes, they study its interactions with the membrane and could resolve one of the states to below 4 angstroms bound to lipid membrane. So very exciting there too. An example of an ATP synthase, and the most exciting example is again ACRB, solved in liposomes at below 4 angstrom resolution. So definitely also a method to look into. The only thing to be aware of here is that in order to produce these thin samples we need for Cryo-EM, you would typically aim to make small liposomes with large curvature, which may or may not affect the structure and function of your target protein. But overall, you know, the field of Cryo-EM and going and being able to solve high resolution structures, you know, is just amazing. If you look at the numbers from, you know, the first structures, you know, more than almost 20 years ago to now, you know, in 2019 or even in 18 already, we solved more higher resolution structures than the lower 4 to 10 angstrom resolution structures. And especially for membrane proteins, the first structures in amphipol and detergents, and now seeing all these different membrane protein structures in a lipid environment, no matter what you're using, the MSP standard nanodisc, SaliPro, PeptiDisc, you name it. And even now structures in these native nanodiscs are coming out, and I'm super excited that we have all that. And if I have 1 or 2 minutes left, I still want to mention the great potential of looking at the much more crowded subcellular fractions or cells themselves, and mention 2 examples here. Here on the left, you see isolated ER fragments, and being able to use cryo-electron tomography and subtomogram averaging and classify and classifications to get quite good resolution information on ribosome docking sites, for example, here. And here on the right, where they even looked at thin areas of intact cells using template-free classification to pull out maps of different membrane protein complexes in such a crowded, natural environment, you would say. So definitely a method to watch, follow, and use, and hopefully we can push the resolution even a little bit further to see more details. And for samples that are really too thick to image, I mentioned that these thicker samples can now be thinned down to so-called cryo-lamella, which can then be used to image or do tomography and use subtomogram averaging. And here I'm showing just the 2 most or best known examples of membrane proteins so far. One is the translocon bound by ribosome here on the left, and the nuclear pore here on the right. So super exciting. Again, Cryo-EM, is so versatile in what you can look at, and it's really time now to make our membrane proteins a little bit more happy by using at least one of these lipid membrane environment systems to look at our structures. And if you're looking for a postdoc position and are interested in cryo-EM or tomography of membrane proteins in a lipid environment, feel free to reach out to me. I just started my lab, and it's located on the main NIH campus. And we have multiple screening scopes, data collection scopes, various computing resources that you can possibly want and need, and newly renovated lab and office space available to us. And of course, a fantastic collaborative environment here at the NIH with access to a huge variety of basic and clinical research. So if you're interested, just shoot me an email and then we can talk about options. And yeah, with that, going back to my summary slide, and if there are any questions, I'm happy to answer them already.
Thank you, Doreen. So I have a few questions here. What I'm going to do is we'll start with one, and I would ask that perhaps while the other panelists are speaking, you may be able to type answers to some other ones. So Sylvie has a question which is around, there are several steps. You have to purify your protein or express it. You have to make the nanodiscs. There takes time. So she has questions about the best ways of preserving protein integrity and prevent degradation. And again, with the mindset that whatever we're doing to preserve this is good for cryo-EM.
Right. No, that's a terrific question. And I know how long it takes to purify a sample. And again, the traditional way of once you have something expressed in larger quantities that you can detect, you usually purify a membrane and then use, screen for different detergents to solubilize your protein and hope that it doesn't just fall apart and aggregate and end up in your pellet. I wish the native nanodisc technology was already so advanced that I can just tell people, go to that company, buy that polymer and prepare your membranes and add these polymers and pull out your protein. It should be fine. We are just not there yet, and my lab will be testing a lot of different available polymers and synthesize ourselves. And we hope that with that, we can suggest something like that. In the meantime, work with mild detergents and try to immediately add some lipids back and do this reconstitution method into nanodiscs, you know, sooner than later, that the chances of your protein, you know, falling apart or destabilizing is lower. So there are, and I can try to you know, be more specific in my answers if they are, or reach out to me, and I can try to share standard protocols that I usually start with when looking at a new protein.
Great, thank you, Doreen. There are a number of questions. We're up to 10, 11, it keeps growing. So, but I think I would like to move on to Maofu, and if you could look at them online and start writing some answers to them, that'd be great.
I will.
So, Maofu, if you could go up next, that'd be great. And again, at the end of this, we can stick around and answer more questions after everyone's had a chance to speak. So thank you for everyone. Keep writing questions. We've got them.
Great. All right. Thank Brian for the introduction, and also thank Protein Society for the opportunity to show you our recent work. Doreen has given very nice introduction of the general nanodisc and also how to study protein in native lipid bilayer. So my talk will mainly focus on application and actually using quite standard lipid nanodisc. And the next talk from Mamu will really show you some fancy stuff. Cryo-EM is a method that generating 2D projection image of biology sample embedded in a thin layer of witcher's eyes. And these 2D images representing individual protein particle in certain orientation has to be determine their orientation computationally, and they will get back project and average in 3D to improve signal noise ratio. So in that way, if the sample is homogeneous, then you can bring back the high resolution information. So the method has been around for decades and have great potential compared with more traditional structural method, but it's not until 2013 when direct electron detector and probability-based 3D classification were implemented in the pipeline. So this make at least single particle cryo-EM widely applicable for high resolution structure determination. Around that time, I was actually doing my postdoc work with Dr. Yifan Chen at UCSF. And together with my friend Hu in David Julius' lab, we determined the first membrane protein structure using single particle cryo-EM. So that's TRPV1, and that's the protein in amphipole, so not really in lipid bilayer. So I will talk more about that in a couple of slides. So at that time, both Yifan and me are super excited and think that's finally comes the time that we can use a structural approach to study high resolution information of membrane protein in the bilayer. So the method has been very well explained by Doreen, so I will skip this. Just a little kind of story. So after this protein structure in amphipole was determined in end of 2013. So they go on to work on the nanodisc embedded TRPV1 structure, and finally published that as 2016. So to my knowledge, that's the first nanodisc embedded membrane protein structure. In 2014, I moved to Harvard Medical School and started my own lab. So my first project is actually try to determine the structure of MSBA in nanodisc. And that took us about 3 years to accomplish. Compared with TRPV1, which is about 300 kD contains 24 transmembrane helices and have a C4 symmetry. So MSBA is about 130 kD, contains half amount of transmembrane helices. And although it's homodimeric, but because the substrate has no symmetry, so we actually cannot apply symmetry during the processing. But more interesting comparison is the general structure of TRuB-V1 is actually almost identical in Amphipole and in Nanodisc. While MSBA in Nanodisc is completely different from the previous determined structure in Detergent. So that's basically one of the main topics I will tell you about. So I would kind of back up one step. Now I will tell you the system we are interested in and also the way we look at it. So here is lipid bilayer. For the substance to cross the membrane, it's often not spontaneous. It requires membrane protein to facilitate the process. And often it's even require energy. And one of the convenient energy resource is ATP in the cytosol. By cleaving the high energy bond and generating ADP and phosphate, this energy can be released and used to push the substrate across the membrane. And ABC transporter use a special ATP binding cassette to bind and hydrolyze ATP. More specifically, this cassette contains 2 nucleotide binding domain, or NBD. So at the interface, it contains 2 ATP binding sites. Usually, they are both active, but in some other case, one of them are active when one of them bind ATP but cannot hydrolyze it. So the binding of ATP will drive the tight dimerization of NBD, and this dimerization through NBD is a very well conserved process, and NBD domain structure and mechanism has been well studied. So the dimerization of NBD will induce the hydrolysis of ATP, and the resulting ADP and phosphate cannot hold the dimer anymore. So the NBD will dissociate from each other. And as you can imagine, cycles of NBD dimerization and release is actually providing a mechanical movement to drive, which can be used to drive the substrate translocation. And then how does it do it? So in fact, within the membrane, there are actually 2 domains called transmembrane domain, or TMD. Unlike NBD, which is highly conserved, TMD is very divergent, which is consistent to their very different functions, which we'll talk more about. Again, ATP binding will drive the dimerization of NBD, and actually such conformation change will propagate to TMD, and often it results in a completely different protein conformation. And these 2 conformations are often referred to as inward facing and outward facing. During its function, ABC transporter basically cycle between these 2 conformations, hydrolyze ATP and transport substrate. So at least this is the paradigm a few years ago when we started. However, the conserved NBD function and structure, and also the paradigm like this, are actually not sufficient to explain the mechanism of highly diverse ABC transporters. So here's the overall picture. ABC transporters form one of the largest membrane protein families, and number-wise is probably just second to GPCR. So it uses the energy from ATP hydrolysis to transport a wide variety of substrate. It pretty much everything you can think of, from iron, sugar, amino acid, lipids to peptide and protein. To understand such enormous of variety is actually, we have to look things in two levels. One is different ABC transporter have very different substrate specificity spectrum. Some of them are highly specific, and some of them is actually not specific at all. Second is they have very different translocation pathway. So at the least, we can consider the entry point and exit point for the transporter at least can be a 4 position, such as cytosol, inner membrane leaflet, outer membrane leaflet, or actual membrane space. And on top of that, transporter can be either exporter and importer, depending on their moving things away from the cytosol or into the cytosol. So there are actually 2 levels of complexity convolute together. One is substrate spectrum, and the other is transport pathway. So they come together, form the enormous diversity of ABC transporter family. However, generally the mechanism, the underlying mechanisms are not super clear for most of the ABC transporter. So my lab is very much interested to perform a mechanistic study of ABC transporter. Specifically, we want to understand substrate transport interaction during the transport. And we want to understand how protein conformation transition can couple substrate movement to ATP binding and hydrolysis. So I will mainly use 2 examples to illustrate how we do this kind of studies. And both of these transporters are actually in the same pathway for LPS biogenesis in gram-negative bacteria. So here is a schematic of the double membrane of gram-negative bacteria. In addition to the inner or cytosolic membrane, it contains the outer membrane with the periplasm in the middle. In the outer leaflet of outer membrane, a very large and complex lipids called lipopolysaccharide is the main lipid component. It's actually have a very important function for bacterial growth, antibiotic resistance, and also induce innate immunity in the host during bacterial infection. The major part of this molecule is called lipid A. So it's a disaccharide, glucosamine, connected to 6 lipid acyl chain. So roughly you can consider that's like 3 regular lipid standing side by side. On either side of the disaccharide, there is one phosphate group. And then it has a decoration of a very long polysaccharide. So one thing to note here is this molecule contains a very large hydrophobic portion, and then is a very large, another part is very large hydrophilic portion. So this complex molecule is first synthesized in the cytoplasmic side of cytosol, or of inner membrane, where ATP and building blocks are available. Then it gets flipped to the other side, and then get mature through a number of modification, including addition of polysaccharide. And then it needs to be extracted from the inner membrane, push across the periplasm, and finally inserted into the outer leaflet of the outer membrane. This very long journey was started with the help of an ABC transporter called MSBA, which flipped LPS from one side of membrane to the other side. So MSBA is actually a long-standing protein sample for membrane protein structural biology. Many years ago, Jeffrey Chang's group has generated a set of MSBA crystal structure in detergent, including 2 inward open conformation with different opening, and then 1 outward open conformation for both ADP vanillate and AMP-PMP bond. Although none of these structures show LPS substrate, based on this structure, they propose an alternative assessing or alternating assay model in which LPS is proposed to be bound in the inward conformation and then released in the outward conformation. However, without actually seeing the substrate, the mechanism of MSBA remain mysterious. So we don't understand how this transporter can accommodate such a large complex lipid, particularly the lipid have both hydrophilic and hydrophobic portion. And really the ultimate mystery is how does this lipid undergo 180-degree rotation? So we take the approach by combining lipid nanodisc and single particle cryo-EM. The first thing we look at is after we incorporate MSBA into a variety of nanodiscs and compare their activity in hydrolyzing ATP with the MSBA in detergent. So we found when MSBA is in nanodisc or liposome, their activity is much higher than in detergent environment, suggesting that the lipid bilayer environment indeed indeed can support the best activity and very likely will preserve the original conformation. So we finally select E. coli polar lipid and also regular MSP1 for our structure study. So we determined the first MSBA structure in nanodisc, and the general folding is the same as previous determined crystal structure. One interesting thing is it contains a domain swapping feature. For example, here one transmembrane domain contains 4 helices from one subunit and 2 helices coming from the opposing subunit. However, the conformation is definitely different. And at that time, we start to think whether the difference comes from different lipid environment, because now we are using nanodisc and we are not using detergent, or that's because the substrate interaction. So, so we'll come back to this question after I basically present all our results. And I think the final answer is likely the difference is actually coming from the LPS interaction. So in our Cryo-EM structure, we resolve a co-purified LPS molecule. It shows the acyl chain, glucosamine, and inner core of the LPS. And the orientation and position of this LPS molecule is very informative. LPS originally present in the inner leaflet has traveled deeply inside the transporter toward the target leaflet, actually with its acyl chain already reaching the target leaflet. So we actually capture intermediate state during the transport. The only thing the LPS molecule hasn't accomplished is actually the 180-degree rotation. So this suggests the entire flipping process is a translation prior to rotation. And interestingly, a recently determined type 4 p-type ATPase phosphorylserine flippase also indicate this lipid hasn't completed the 180-degree rotation when it's already reached the target leaflet. So translation prior to rotation may be a general scheme for ADP-powered lipid flippase. At the level of phosphorylated glucosamine, we observe a ring of arginine and lysine surrounding the two phosphate. So they form a highly stable electrostatic interaction. And in the interface, above the glucosamine, there is a large hydrophobic pocket accommodating the 6-lipid acyl chain. So taken together, electrostatic interaction around glucosamine and hydrophobic interaction accommodating the lipid acyl chain are the structural basis for specific MSBA-LPS interaction. In contrast, the oligosaccharide extending into the cytosol seems far away from the protein and seems unlikely to mediate specific interaction. So next, I'm going to use a movie to summarize our finding. In the bacterial inner membrane, MSBA originally adopting flexible conformation, and the trapping of LPS stabilized the conformation, and that's represented by the structure we determine using Cryo-EM. And it also promotes ATP to bind at the interface of two MBD. The binding of ATP will drive the tight dimerization of nucleotide binding domain and convert the conformation of transmembrane domain, and likely open up the distal end towards the periplasm. So we were not able to use Cryo-EM to capture this conformation, but it's suggested by previous crystal structure. We think this could be an important intermediate to expose the hydrophobic acyl chain to the aqueous environment of in periplasm, which will generate the direct driving force to rotate the lipid acyl chain to hide in the neighboring membrane environment. So after LPS is flipping out and ATP get hydrolyzed, the inner cavity of a transmembrane domain will collapse. So we also determine the structure of that conformation state, and later we improve the resolution by using AMP-PNP to trap that state. After release of gamma phosphate and ADP, MSBA reset to the original conformation and ready for the next cycle of transport. Here is basically a static version of the movie I just showed you. We call this model trap and flip. There are two steps we still know relatively little about. First is initially how LPS get trapped into the transporter. And second is exactly how LPS flip out. Related to the first question, our most recent unpublished result shows that with the under certain condition, we were able to generate similar wide open conformation in lipid nanodisc. So the lack of membrane environment probably cannot explain the wide open conformation, and such conformation could be functionally relevant. But that would require more future work to confirm. So now I'm going to switch gear to talk about another ABC transporter, which downstream in the LPS transport pathway. It has, compared with MSBA, it has a function, quite different. So instead of flipping LPS, it's extract LPS out of the membrane and deliver to a protein acceptor in the periplasm. And folding wise is also different. Unlike a homodimeric MSBA, this transporter has 4 individual protein called LPTF, LPTG, and 2 LPTB. So we took a very similar approach to determine LPD-BFG structure in Nanodisc. And here you can see the transmembrane domain from LPTG and transmembrane domain from LPTF with 2 LPDB in the cytosol. We were super excited to see this structure actually resolve a co-purified LPS density. An interesting comparison regarding Nanodisc and Detergent is almost at the same time, 2 crystal structure of LPD-BFG were published from different species, but none of them resolve clear LPS, except one of them actually show clear extra density. So it seems like here is a situation similar to MSBA, where LPS is very likely co-purified at the beginning, but either lipid environment is essential to keep LPS stably bound, or LPS simply released during the long process of crystallization. So now back to the Cryo-EM structure of LPD-BFG. The LPS density clearly resolve inner core phosphorylated glucosamine and lipid acyl chain. Our structure even clearly resolve the secondary acyl chain connection. So such quality actually allow us to assign every individual lipid acyl chain. So this lipid acyl chain packed tightly in a protein pocket. And we have the sidechain resolution to demonstrate the close interaction with the lipid acyl chain. And many of these sidechains has been mutated to prove their function in bacterial growth and LPS transport. At the level of phosphorylated glucosamine, again, we actually see a ring of lysine and arginine interacting with the phosphate. But there is an interesting difference compared with MSBA, is most of these positively charged sidechain coming from LPTG, while only a few coming from LPTF, indicating these 2 proteins, although they are structurally very similar, their function for LPS transport is not symmetrical. Now, comparing MSBA and LPT, we can see they both use electrostatic interaction to interact their glucosamine, and also a large hydrophobic pocket to accommodate the lipid acyl chain. However, they will arrange these elements differently in different position and also in different orientation, so that these 2 transporters can fulfill their very different function. One is flip LPS and the other is extracted out of membrane. Okay, so here is not the full picture. So very briefly, so it actually, the cross periplasm transport require a set of 7 proteins called LPTC, LPTA, and LPDD and E. So our further work look at the interaction of LPDC and the minimum ABC transporter. Compared with DDM, these 2 complexes show much higher ATPase activity in Nanodisc, and very interestingly, the one without LPTC is actually showing higher activity, suggesting the full complex is actually self-inhibited. So the reason become more clear after we solve the structure. LPTC helices is actually between the 2 transmembrane domains, and it's probably inhibiting the conformation change, which is required to hydrolyze ATP. And we directly proved that by determining the structure of vanillate trap complex, showing that these helices have to be removed away from the interface. So here's the model, which I will skip, but the key take-home message is we reveal an unexpected critical role of LPDC to sense the presence of LPS and blocking the conformation change without the binding of LPS will prevent the futile ATP hydrolysis. So far, we look at 2 ABC transporter, and very recently, we also determined the structure of another ABC transporter called LLCDE, which is transport lipoprotein in gram-negative bacteria. And this 3 protein is actually the only 3 essential ABC transporter for E. coli bacterial growth. So we are also working on pharmacological regulation of these transporters, and hopefully can contribute to the development of novel class of antibiotics. And we also extend our work to human ABC transporter, which I will just skip. So in summary, the combination of lipid nanodisc and single particle cryo-EM allow us to study ABC transporter in lipid membrane environment and also in the context of substrate transport. So we are really in the early stage to understand how different ABC transporter can transport a different thing in a different pathway. So here's I would like to acknowledge my previous postdoc who did most of the work, and also our facility and the funding. Thank you all.
Thank you, Malfu. So we have a couple questions here we could get to quickly. Okay. Someone asked, so his name is John. This is basically the MSBA structures. Do you use N-terminally tagged proteins? They're commenting on that these tags could perturb the ATPase activity.
Yeah, we do. We use the original construct as the previous crystal structure. So it is N-terminal. Later, I think we changed the tag also to C-terminal. I need to look back to see whether we actually carefully compare the ATPase activity, but that's a good point. Thanks.
Okay, great. And this is coming from Rodrigo. Do you think Cryo-EM can only show limiting steps where an intermediate accumulates? Like, I guess it's about how many steps in the chain can it really capture?
Yeah, I think I like to think about that as essentially, if there's multiple steps, eventually which step actually accumulated in a snapshot depends on which step is actually the most stable and the step which stay for the longest. So if there is a step which much longer than all other steps, then if we do such analysis, then we will just only getting one state. I hope I'm answering the question correctly. So I consider that is asking about the continuous reaction.
I think this is good. You can see the questions too written down. So I think it's good that we move now on to Mahmoud's talk if you're finished.
Yep.
And if you could just look at the questions that are written down, a couple of them have come in for you, and just write some answers to them. We can move those forward. And again, afterwards, we can get to things that haven't been answered yet. So thank you for your presentation, and we look forward to hearing from Mahmoud.
Trying to unshare.
Okay, there you go.
Okay, thank you very much, Brian, for the kind introduction. Thanks. I would like to thank the Protein Society for the invitation. Today, I'll talk about Cryo-EM structure of an activated GPCRG Y protein complex in lipid nanodisc. But first, I would like to acknowledge people in my lab, the current and former people in my lab, as well as our collaborators, especially in the Wagner and Brown lab. And more importantly, special thanks to Mang Zhang and Miao Liu. They are two outstanding postdocs. Without them, this wouldn't be possible, especially the Cryo-EM work with the GPCR Nanodisc. So my talk divided into 4 parts. The first part, I'll talk to you about covalent circularized nanodiscs and mention briefly the development of the DNA corralled nanodisc. And next, I'll talk about why study GPCR in bilayer, the importance of the bilayer. Next, I will move on to the preparation and cryo-EM structure of Neurotensive receptor in G protein trimer complex in nanodisc. So just to remind you, ApoA1, the nanodisc inspired direct from nature. The liver makes that ApoA1 protein. This ApoA1 bind to lipid in blood and then form that pre-beta HDL. Pre-beta HDL continue to accumulate cholesterol from peripheral tissues and matures to alpha HDL. Alpha HDL then taken by the liver through the interaction with the receptor, the scavenger receptor class B1. So ApoA1 itself can form nanodisc, and several labs used it to make nanodisc. The MSP1D1, which is it's widely used in biochemistry and cryo-EM. It's basically it's derived from ApoA1 by removing the globular region of ApoA1 and half of the helix 1. So Zweigart did beautiful work incorporating different membrane protein in these nanodiscs. Also, he designed constructs such as MSP1E3D1 and MSP2N2, and have been used extensively by the cryo-EM community to solve many structures. So I was interested really to expand the size of nanodiscs beyond the 16 or 17 nanometers that we can be accomplished by the traditional nanodisc. Also, I wanted to, we're very interested to improve the homogeneity of nanodisc and stability as well. So we designed several constructs. This is the general outline of the constructs we designed. So basically, we used sortase to link the NNC termini of MSP. We added LPXTG or sortase recognition site near the C terminus. And here you have glycine near the N terminus. First step, we add TIV to cleave the hashtag to expose the G, and then we add sortase. So the NNC covalent, the NNC linkage ensure really that the nanodisc, the MSP that used in preparation of nanodisc would form homogeneous nanodisc if, of course, attention given to details and removing of the byproducts of the circularization. So these nanodiscs showed improved homogeneity, thermostability, as well as proteolytic resistance. So as expected, the covalent circularization improved the thermostability. To the left here, you see the stability of MSP before and after circularization. On the right, you see the stability in presence of lipid before and after circularization. Also limited proteolysis if you subject the conventional nanodisc to proteases. After 3 hours, you can see here about 60–70% of the protein is degraded. On the other hand, for the for the circularized nanodisc, up to 3 hours, nothing happened really. And next day, after 18 hours, you can start seeing the band above it's around 22 kD. This actually represents the opening of the circularized protein. So we designed several constructs, including NW30, which assembles 30 and 15 nanometer. Also, we designed NW50. NW50 assembles about 50 nanometer nanodisc. And actually, I wanted to quickly share with you how we purify it. The reason for that, because this is the most difficult construct we have in terms of purification and stability. So the first step we add TIF, as you can see here, before and after TIF, you can see the shift. And then we subject the protein to size exclusion chromatography. We collect the fractions enriched with the full length, not the truncation products. As you can see, a large truncation. And then we subject it to another round of nickel purification, and eventually perform circularization. If you really jump from here to circularization, you end up really having a lot of contamination and byproducts, and will end up having low quality nanodisc. So we made these large nanodiscs, and we're excited about them because we start using them to study viral entry and interaction with bilayer. So this, just to give you an example, this is the 50 nanometer nanodisc in vitreous eyes. You can see here clearly, this is the polyvirus. It's a non-enveloped virus. And you can see it's dark because the RNA is still inside. You can see the nanodisc here, the 50 nanometer. This is a tilted view of the nanodisc. And right here, you can see actually the viral particle sitting in the top of nanodisc after ejecting its RNA. You can even see the egress of the site of RNA egress. So we use these large nanodiscs to study really how the non-enveloped and enveloped virus interact with the bilayer. And we wanted next to study the genome translocation and the entry intermediates at high resolution. So for the smaller nanodiscs, the smaller covalent surprise nanodiscs, including NW9, NW11, we use them to incorporate a variety of membrane proteins, including ion channels such as VDAC, transporters, as well as several GPCRs. So another type of nanodisc is DNA coral nanodisc or DNA belted nanodisc. So the way we make that is to start with the DNA origami and actually attach a conventional nanodisc to the lumen or the inner side of the DNA. And then as you add more lipids, you actually assemble the bilayer inside the nanodisc. So the reason we actually were interested to use DNA is to help stabilize these large nanodiscs and go beyond 50 nanometer. Here you can see the EM images for the DNA, the ring itself, before adding the small conventional nanodisc. You can see here, these are the small nanodiscs after decorating the inner face of the DNA organ. So this is what we think happened step by step. So you can here see the smaller nanodisc as you add more lipid, local fusion happened between two neighboring discs, and then eventually you end up constructing this large bilayer, which allows you to really study virus entry at high resolution. This is just to give you an example and feasibility of using these DNA coral nanodiscs in studying virus entry. You can see here, this is a polyvirus. And actually what you see here, you see side view of the bilayer and left behind it the DNA ring. So you can see actually nicely the bilayer, and you can see different views. Interestingly, we can see actually the virus bending the bilayer and also forming putative pore here through which we think RNA go through. So also we are very interested in actually not only forming circular nanodisc, we are interested to develop polygonal nanodiscs and manipulate the shapes. For example, we can make triangle, rectangular, hexagonal nanodiscs. And we actually successfully made elliptical and polygonal nanodiscs, small ones, by actually strategically modifying and inserting amino acids, cysteines, at defined location. When the build protein fold over itself, it actually formed disulfide bond. So initially we performed the nanodisc assembly in presence of reducing agent, and as we dialyze out the reducing agent, we end up forming the disulfide bonds. These nanodiscs extremely stable and has potential applications in like diagnostic and other applications that requires really very stable nanodiscs. So we make these stretched hexagonal nanodiscs, and are very excited about that. Of course, other potential of using these polygonal nanodiscs include membrane crystallization, membrane protein crystallization, Cryo-EM application. Also, we can you can stitch them together and build 3D shapes, including like a pyramid shape or virus-like particles for different applications. Also,
Mahmoud?
Lab is also creating asymmetric nanodiscs. Next, I'll move on and share with you my thoughts why we really have to study GPCR and bilayer. So to quickly give you a brief introduction, so once the GPCR binds to its ligand, that binding induce changes in conformation that favor binding to the G protein heterotrimer. And then this binding results in actually, this binding stimulates conformational changes in the alpha unit that results in the GDP release and GTP jump into between the RAS and alpha helical domain. And then following that, the beta gamma separate from the alpha and then mediate downstream signaling. Most of the structural studies for GPCR G protein interaction required various methods of stabilization, including using antibody engineered dominant negative G alpha, engineered mini G protein that lack the alpha helical domain. Also, the majority of the structural studies of reconstituted GPCR complex is done in detergent micelles. One important thing to mention is of course, a lot of literature shows that the lipids can modulate receptor functions such as ligand binding, receptor activation, G protein binding and coupling, recruitment of GPCR, kinases, and arrestin, to just list few papers. Here, this is a report from Carol Robinson group, shows that PIP2 stabilizes the active states of GPCRs and enhance selectivity of G protein coupling. Another report from Koberka group shows that the local movement charge regulates beta-2 receptor coupling to GI. So we really believe that structural and dynamical information for GPCR protein interaction and a lipid bilayer is necessary to understand the GPCR's signal transduction mechanism. So next, I'll move on to the preparation and characterization of neurotensin GI complex encirculated as Nanodisc. For this study, we chose this construct of a neurotensin. It's called TM86V L167R. We obtained this construct from Andreas Klopfen group, and this construct exhibits most wild-type-like activity and native signaling function. Also, we used in this study the wild-type heterotrimeric G protein, the G alpha I1, beta 1, gamma 1, which is myristoylated on the gamma alpha 1 and preannihilated on the gamma subunit. So this is how we made it. So basically, the neurotensin was purified using the neurotensin peptide immobilized on beads, and then incorporated into circularized N9, which assembles at around 9 nanometer nanodisc. And that incorporation actually resulted in enhancement in stability of the neurotensin as compared to detergent. Interestingly, if you look here at the binding between the neurotensin and GI, you can see enhancement of affinity as a result of incorporation of neurotensin receptor in nanodisc. So you can see the KD right here is 76 nanomolar for the GI against the neurotensin in nanodisc versus 1.4 micromolar for the neurotensin in detergent. Here you can see BLI signals, and the BLI signals show the dissociation between the GI and neurotensin in nanodisc. In absence, here you can see the black and brown, and in presence of GTP gamma S, showing faster dissociation of the complex in presence of GTP gamma S. And this suggests the ability of the neurotensin GI complex in circularized nanodisc to perform GTP exchange. We also measured affinity in different lipid using neurotensin in nanodisc made of different lipid composition, and we found that including negatively charged lipids such as POPG enhances the affinity. So next we went on and we made the complex with the GI. So first, after the preparation and purification of the neurotensin in nanodisc, we added the GI to the nanodisc, and then this followed by adding a pyrase. By adding a pyrase, you really remove or degrade the GDP, and then you stabilize the complex. And then we run SDS page to confirm the incorporation of all the components into nanodisc. This is an MR done by Mangjan, and you can show here the neurotensin and nanodisc. You see the peak well dispersed and nicely resolved. And also we see that these nanodiscs are stable at 45 degrees for 15 days at least. And actually, after you're adding the GI to the neurotensin nanodisc, you can actually see shifts in many peaks indicating binding of the GI to the neurotensin. So next we went on to do the Cryo-EM structure determination. So the first thing, of course, we do do negative stain, and then as you can see here, the sample looked really nice, and we were ready to move on to the Cryo-EM. These are Cryo-EM 2D class averages, and you can see already the secondary structures, and you see nicely the density from nanodisc here. So what you look in here, you look at intracellular view, you can look at the beta, gamma, alpha, and you can see the density of the nanodisc behind here. And here, for example, this is a top view from outside the cell. You can see actually the neurotensin receptor, and behind it, you can see the G protein tribe. And this is side view. So this is the workflow, and this is how we processed our data. So we actually ended up having two structures, canonical and non-canonical states. This is the local resolution map. And interestingly, the pitch and many side chains are clearly resolved, allowing us to place and remodel known atomic models of neurotensin and GI. And we can also see clearly the density of the neurotensin peptide. On the top here, you see the canonical state, and in the bottom here, you see non-canonical state, and they are related by the 50-degree rotation of the GI around the neurotensin receptor in nanodisc. We can see a lot of lipid interactions. For example, you can see here, this is the alpha N of the G alpha. You can actually see the side chains of arginine and lysines protruding upward, interacting with the negatively charged nanodisc. Here you can see actually the myristoylated and premyelated modification of the G gamma and alpha, also protruding and interacting with the nanodisc. Here is a comparison between our structure and other structures in Detergent, and as you can see, in case of canonical state, you can see actually the alpha N is very close to the membrane relative to other structure. Also in non-canonical states, it's very close. We're also we're able to see the alpha helical domain, and in our structures, the alpha helical domain moves away from the RAS domain to interact with the second and third blade of the G beta. And we think this is really reflect the native, more of a native position of this. For the few structure that we're able to see density for the alpha helical domain, these structures actually, again, they used Fab and nanobodies, and these the position of this alpha helical domain may be affected by the crystal contact or the nanobody antibody that were included for the stabilization. Here we see actually interestingly that the core of the neurotensin is more compact due to an inward movement of middle of TM6. And we think that the bilayer constrains the conformation of neurotensin to enhance its interaction with the GI. Here you can see the interaction between the neurotensin and the alpha N of the alpha unit. You can see actually more interaction in the canonical state versus the non-canonical state. What you can see here, this is a potential salt bridge between glutamic acid E28 and R185. Also, we see several hydrogen bonds between E28 and S182, and between arginine 32 and T179 right here, as well as the alanine 31 and lysine 178. Also, the alpha 5 of the alpha domain show more interaction in the canonical state. In contrast, we only seen one hydrogen bond in case of the neurotensin receptor and detergent structure. So this is just to overlay the structure in detergent versus structure in nanodisc. You can see here clearly the upward movement of the alpha N of the alpha, and you can see several changes in the structure here. So we really think our structure provided molecular explanation for the stronger interaction between GPCR and G protein in bilayer. So to summarize, we determined the structure of neurotensin receptor in complex with the G alpha I in two conformational states, canonical and non-canonical, and the structures were determined without any stabilizing agents. And our structures not only highlights difference with the GPCR G protein complexes in detergent mice, but also provides a molecular explanation for a stronger interaction between the GPCR and G protein in lipid bilayers. And we've done a lot of work we use BLI and MST to show these affinity differences. And at the end, I would like to thank you again, and I'll be happy to take any questions.
Thank you, Mahmoud. Great talk. We have some time here, so I do have some questions that I think you'd be really good for answering for sure. So there's a gentleman, or I don't know for sure, but I'm saying, forgive me if I say the name wrong, Zainab. So they're working with integrin receptor. So in the inactive form, it goes into the nanodisc, but when it gets activated, it's not associated with the nanodisc. So there are questions about how to get this type of proteins integrated into the nanodisc that I guess in one state it does, and in another state it does not.
I mean, some explanation, again, I don't know about this protein, but some potential explanation, the one conformation maybe more prone to aggregation or oligomerization that doesn't fit to the nanodisc. I would try, for example, different sizes of nanodisc, maybe use bigger one. It's really hard to, you have to just try different sizes, different lipid compositions, etc.
Great, thank you. Another question is, do your large nanodiscs generally contain significant cholesterol? Are there preferences or limits in terms of lipid type?
No, we made these large nanodiscs using different lipid mixtures. You can definitely include cholesterols, and we have already. So yeah, depending on the target you're studying and whether cholesterol is essential or not, so we can include it. Yeah, so case by case.
And this one's regarding the NMR spectra. What is the status of making chemical shift assignments for the two NMR spectra? What kind of nanodiscs are used?
So Mong Zhang currently working on this, I believe, and she made progress. I believe she's using the NW11. Okay. Right. So yeah, and we're doing this in collaboration also with Franz Hagler and TUM. Yeah, but I think there's a lot of progress made already.
Great. Let's see here. Okay, this is another question. Does the mutants, or do the mutants have similar changes in affinity for Ntrga interactions in detergent versus nanodisc?
Yeah, it has a higher affinity to the GI when we place it in nanodisc. It's 76 versus 1.4 micromolar, 76 nanomolar versus 1.4 micromolar in detergent. And as you can see from our structure, we see that the alpha N of the G alpha interact tightly with the membrane, and there are other membrane interactions as well.
So the next one is the MSP belt. Is it ever a problem during electron microscopy tomography where the MSP gets in the way of the target protein?
I don't know.
Okay. I guess there is time, you know, this is scheduled to go till 2 o'clock. So I think we can open up, if Malfu, if you're there, and Doreen, we could go, and there are several questions still on deck. We can open it up to a general discussion. Let's see here. Okay, this was a good general question. So in regards to, and any of you guys can tackle this, so the minimal size of proteins, this is where around, what are the limits of cryo-EM in regards to protein size? So the question is, the person's working with a 25 to 30 kilodalton protein, and I'm assuming it's a monomer. So I guess we could approach it from monomer, you know, and what are the limits of size needed, and where is the field at right now? I guess Doreen, if you could do that, or Malfu, anybody? Everyone's on mute, so if you get off.
I think Doreen probably know better about the literature. So what's the current record of the
Sure.
smallest protein in nanodisc?
Yeah, the smallest protein in nanodisc. I mean, Mahmoud, I mean, your GPCR, just the membrane part, is pretty small, right? I think that's probably one of the smallest ones, and then you just increase the size by adding all your additional proteins that bind to it. So what's the size of just the membrane part, for example, in your case?
So the membrane protein is about between 30 and 40 kD, right?
Right.
And,
So
the G protein, the alpha, beta, gamma, roughly at 100 kD.
yeah.
Yeah.
Yeah. And I mean, it still has to be shown whether without these additional soluble, you know, binding proteins, this protein can be solved without it, because all these additional proteins, they help you align your protein complexes when you process your data. So that still has to be shown. But the moment you probably add a fat fragment or something to something that's fully inserted in the membrane at the size range, 30, 40 kilodalton, that's where we're at right now. And it's all about pushing that limit a little further. And the 25 to 30 doesn't sound too far off, but it's going to be challenging.
So the biggest challenge is proper placement or arrangement of your protein based on the data you're collecting. Can you just describe for them, like what's the limitation that's hard for cryo-EM as it gets smaller?
Yeah, so when I showed you my cryo-EM image, or if you looked at the cryo-EM images from all of us, you're really looking for some signal inside these either nanodiscs or detergent micelles or what it is. So if you have a thin enough sample for cryo-EM where you can see your particles and you even see signal inside your membrane part, there's a good chance you can align it. But it's really in terms of getting there to get your small sample into this thin ice layer, really nicely distributed and still seeing signal inside that disc. Once you see that, and you can align it, but the smaller it gets, the larger the smaller the signal to noise ratio is. So and that's really, you want you want a high signal to noise ratio to be able to align, all your particles together into an average structure.
Okay, great, thank you. So here's a general question. How do you determine the ratios of MSP, lipid, protein for each case? And how do you choose the MSP to be used? So it's more of a basic research question. How do we optimize? What kind of grid do you start with? How many conditions?
We usually do a few parallel experiments to test a range of ratio. So generally, we feel for the standard MSP1, I think 1:60 or 1:80. And also that depends on lipid too. And mostly our lab use, most of project we use POPG. So 1:60 or 1:80 seems can give us the best result. And for E3, it usually go to 1:120. and N2, it could go up to even higher. A general thing we observe is with smaller MSP, definitely it's more homogeneous. And with larger one, we will have kind of more population, more distinct population of different size. And also that also depends on your target protein. Target protein can actually also induce like a different preference of different size. So we always do kind of a set of different condition to compare.
Okay, great, thank you. And Doreen, this is a real sort of like standard operating procedure question about biobeads. Anybody else has experience that can answer this question as well? It sounds like, so I'm just going to read it out. So using Cryo-EM to study the structure of MTHK, and I always use biobeads overnight after 2 rounds of detergent removal. Would you have any other idea to remove detergent? Because by the time I change biobeads the second time, it's already late at night. And then I leave it overnight for the next day and do gel, size exclusion. So how could I avoid this? Can I restore the protein overnight or store it on ice or whatever? So it sounds like they're running out of time and they run into some negative aspect.
Yeah, I think dialysis is another way, using dialysis.
Okay.
Yeah.
Right. Yeah. Dialysis is a nice other way. And some detergents are harder to dialyze out than others. But if the preference is still to use biobeads and it's at the end of the day, and you really, at the end of the day, you want to go home and get a good night rest, you can always take your sample out. Don't leave the biobeads in and start again the next morning by putting freshly equilibrated biobeads in and just time it a little bit better. Again, I know some people do it overnight and they're fine, but we've really seen the nanodiscs getting more heterogeneous in size, especially smaller. So we believe that the longer you incubate with these biobeads, the more the lipid may actually be taken out, or it leads to these heterogeneous populations. So you want to be careful with the timing, and if you need to start again the next morning, just add a new batch of, if you think it's not complete yet, just add a new batch of equilibrated biobeads in the next morning and time it.
Another thing is the actually using high CMC detergent help remove very low CMC detergent. Like for example, if you have Triton or other detergent that really hard to remove, adding something like sodium chlorate actually help remove it. So we use this trick and it works well.
And I think in the question was also, should I store it at 4 degrees or -80? I'm not a big fan of freezing your proteins in between. So I hope your proteins, where your proteins are stable at. 4 degrees is a good start. If you have screened that your protein is stable overnight at 4 degrees, that's what I would probably store it at. And I wouldn't put it into the -80.
Okay, I have a question for Maofu. So it says, in your presentation, you mentioned that substrate binding in TMDs helps ATP binding. How would that happen, taking into account the relative important intracellular concentration of ATP? Does the transporter keep going into hydrolysis cycles despite the absence of a substrate?
That's an interesting question. So I think for any ABC transporter exposing its MBD in the cytosol, face a challenge or question is whether you want to keep doing it without seeing its substrate. And from all the study we have done so far, ABC transporter seems to have one way or another to lower the ATPase activity when there's no substrate. So in the MSBA case, we have more and more evidence to suggest MSBA actually avoid hydrolyzing ATP by adopting such very large open conformation to physically increase the distance between 2 MBD. And when substrate comes in, it actually closes it up and bring the 2 MBD close to each other. And that promotes ATP binding and hydrolysis. And in other APC transporter, they have other interesting way to do this.
Great, thank you. And this is an interesting question. What are the comparative stabilities of membrane proteins in detergent micelle, bicell, amphiboles, nanodiscs, MSPs, and SMALPS? So I guess if you had a protein and you put the one into each of these environments, is there anyone done this for comparative purposes? You know, for stability comparisons?
Yeah, I think I had a similar question. What's which system gives the highest resolution in Cryo-EM and all that? I tried to answer it. It was like, I wish we had the data by, having a couple of test proteins, putting it in all these different conditions, making the same really high quality grids, that there's no difference between the sample, and collecting data and doing that. Same with stability. I wish we had that data by putting all these different proteins into all these different systems and measuring that. And that's something we need to do, or at least compare. Some labs have a preference for now the peptide discs versus the Salipros versus the MSP. So I'm not aware of anyone comparing all of them yet, and it's something you need to screen and figure out, and that's why meetings and going to hopefully physical meetings again in person, talking to lots of people and exchanging notes on the different methods will be really helpful, and to make some sort of general statement of this has led to longer stability of a protein in that system, or talking about resolution and so on.
Great, thank you. And this is another question around, getting lipid, ratios of the different components. But this one's a little different in that they're asking, when you generally screen, are you approaching this with like a mouth with your parallel? Are they very relatively small scale samples? And then you scale up when you found something that works. And then I guess, or is there another process for that? Or I guess I would add to that, even if you found a small scale condition that works, do you still have to optimize to go to the larger scale? Because sometimes when you scale up, you can lose some of the results change. So
yeah, here is definitely the case. Scaling up doesn't always work. I think as the example of adding BioBeads, I think usually we add a huge amount of BioBeads, and really the small scale and larger scale is changing the kinetics of detergent removal, and that actually changed the result a lot. And one of my postdocs actually mentioned this very early on, and he always do multiple tubes when he finished optimizing small scale. He just do a few tubes.
Yeah. Okay, I think we've done well with answering questions. They're now perhaps 10. And we're closing up. We got 2 minutes left to.
There was one question, if you don't mind, from Mafu.
Yeah. Okay.
So I actually posted that question.
Oh no! It ended up under the answered condition. So let me pull it up.
The MSB transporter, does it flip LPS when it's modified with the O antigen?
When it flip LPS is actually not, strictly speaking, it's not LPS, it's actually LOS, lipo-oligosaccharide. So it only contains inner core and outer core. So it doesn't have the O antigen yet. Right,
but can flip the LPS,
the long version.
A long version of LPS, can it flip it back?
I don't know, because in the cell, it doesn't have a chance to see that substrate. So this has to be reconstituted in certain in vitro assay.
Okay.
Because the addition of that oligosaccharide is actually after MSPA flip the lipid.
Right, so it doesn't go back. you think?
No, it should not go back. I mean, the whole machine is designed for flipping it
One
to
way.
one direction.
Okay. Thanks.
I just want to, I think it's good to conclude now. And I guess each of us, if you could, continue to try and answer the questions that are still posted, that would be great. And I do want to thank everyone here. It's been great working with all of you. And thank you to Raluca and the Protein Society and everyone that's came on for this webinar. It was a really fun time and a lot of good information and good engagements. This was great. So I guess thank you all, and we conclude this webinar, and keep your eyes posted for the next one. Again, you can check all of us out on our websites for the labs and LinkedIn, connect by email. We're all here, so please reach out.
Thank you.
All right,
Thank you.
thanks a lot, everyone. Enjoy the rest of
Bye.
the day. Take care. Bye-bye.